Tuesday. Antivenoms can neutralize the venom or venoms included in their production or closely related venoms. The corollary of this is that it's often a very good idea to have at least a rough idea what kind of snake has bitten a patient so that you can administer the corresponding antivenom. So how do you even figure out which snake has bitten a patient in the first place? Well, there are different types of information that can help with this. One type being the patient history, so the circumstances of the bite, like the time of day they were bitten, where they were bitten. For instance, if the patient was bitten at night while they were sleeping, well, that probably indicates it's some kind of nocturnal snake. In some areas, that alone might indicate that it's a crate. There can also be hard evidence, like the patient or a bystander either capturing, killing the snake, or managing to snap a photo of it that can then be identified by a herpetologist. And finally, there is like the main approach, that is the symptomatic approach. So in the symptomatic approach, the medical personnel will look for clinical manifestations of the envenoming that are characteristic for different types of snakes, and then administer the corresponding antivenom. This approach can be supported by different laboratory methods that detect the effects of the venom, such as the 20-minute whole blood clotting test. However, today I'm not going to be talking that much about tests that measure the effects of venom. Rather, I want to discuss the possibility of developing tests that detect the venom itself. And that's because I believe there could be several potential benefits to having such tests, with probably the main benefit being improved patient care. So I believe that venom detecting tests could potentially lead to rapid diagnosis, which in turn could lead to early and correct treatment. And there are a lot of studies that show that early treatment are associated with a better patient outcome. But how do you get from rapid diagnosis to early treatment? I think diagnostics could help with this in different scenarios. So, for instance, there are some places, such as in the Brazilian Amazon, where snakebite victims sometimes have to travel very far from where they're bitten to a centralized hospital where they can receive treatments. And this, of course, delays the treatment. To counteract this, there are current ongoing initiatives to decentralize antivenom in the Brazilian Amazon, to essentially take the antivenom out of the big hospitals and put it into smaller, more rural clinics. But at least in my opinion, this requires that you have medical staff at those small clinics who not only know how to fill a syringe with antivenom and inject it, but who also know which antivenom to put into that syringe in the first place. And I think diagnostics might be able to help with this. There is also the sort of opposite scenario where patients actually present to the hospital very, very quickly after they're bitten, even before the venom has time to work. But sometimes in these cases, it's not really possible to treat the patient, even though they present early to the hospital, because you can't diagnose them, so you can't treat them. So potentially also in those cases, having something that can detect the presence of a specific venom could help ensure that treatment could get started sooner. So patient care could potentially be improved with better diagnostics. I also believe that diagnostics might have a role to play in epidemiological studies and in resource management. Specifically, venom detection tests could help map risk species and inform decision makers which antivenoms to procure and where to distribute those antivenoms within a healthcare system. And finally, I think diagnostics could be beneficial to essentially a whole treatment life cycle, right from the design phase, where they could help figure out, okay, which are the risk species that this treatment has to cover. And I know I wrote antivenom, but this could really be any treatment. It's like which treatment to cover, to clinical trials where the results from diagnostics could be used as inclusion or exclusion criteria, to like once the product is on the market and you could have companion diagnostics. So there are all these potential benefits of developing diagnostics. But what's actually in the research pipeline? What do we have out there? My colleagues and I did a review on this which was published back in 2021. And what we found was that people have looked into different types of lab tests to try to develop these diagnostics. So there are some enzymatic tests, there's some actually very, very cool molecular biology tests, like there's some nice PCR, but the majority of the research is different kind of immunoassays with an especially high number of ELISAs. Now, to the best of my knowledge, at least, the only such test that's actually currently on the market and in regular clinical use is found in Australia, and it's kind of, you could say, like a rapid ELISA, a modified ELISA system. Also, something we didn't include in this review, because this was more focused on lab-based method, is the possibility of using artificial intelligence to identify snakes from photos taken by victims or bystanders, which I think is also a very cool idea. Okay, but there are a lot of challenges to actually developing these diagnostics. And a not insignificant type of challenge are the technical challenges. So for instance, in my work and my colleagues' work, we're working a lot with point-of-care diagnostics. And we really try to keep the World Health Organization's so-called assured criteria for point-of-care diagnostics in mind. So essentially, that point-of-care diagnostics need to be affordable, sensitive, specific, and so on. But take this sort of requirement or criteria with affordability, for instance. If you want to create a very affordable diagnostic tool, well, then you probably need to rely on a treatment that's affordable, on a technology that's affordable. But that might conflict with, say, having the most sensitive technology out there. Also, in terms of sensitivity and specificity, there's a lot of challenges, just because in many areas there is a huge variation. Like there are so many different snake species present, many different venoms, and sometimes even within the species there can be venom variation. So like, how do you cover all that and have a test that's sensitive for everything and specific for the relevant species? That's quite tricky. There's also something like user-friendliness and robustness, and I'm going to give you an example of that from my own research. So in our research, we work a lot on developing lateral flow assays, and we really want these assays to be as user-friendly and robust as possible. And at least to us, that also means that ideally the test should be able to work with as many different sample types as possible, so that like whatever sample you have available, you can use that and get useful information from it. So in the first iteration of our lateral flow assay, we worked mainly with urine samples, but we also wanted to see how well the test could detect venom in other sample types. So what I have here is just a graph where on the y-axis you have like the sort of a normalized signal intensity. So like the higher the bar, the stronger the signal on the test. And then we have that as a function of venom concentration in different types of sample. And what we can see, especially for the just like running buffer and urine samples, is that, well, when we add a lot of venom, we get a high signal. That's nice. When we add no venom, we basically don't get any signal, which is also good. But then we tried doing the same thing in the serum and 2 different kinds of plasma. And what you might notice is even in the absence of venom, we actually get a signal. So we have false positives. And of course, this is not ideal because you want to be able to tell if it's a true positive or false positive. This particular assay was also not optimized to work in plasma, but it was just something like, okay, maybe we can do this better next time. Another thing we wanted to look into is, okay, one thing is different sample types, but what about just variations within one sample type? Because blood isn't just blood, serum isn't just serum. There can be a lot of different compounds present in samples from different people. So we wanted to try to screen some of these many different compounds and figure out if potentially they could cause false positives. So stuff like different kinds of sugars and salts. We have caffeine in here also, for instance. And when we screened this in serum, we found that there were actually different compounds that did cause false positives. For instance, heparin, which is interesting because heparin plasma also had a lot of false positives, so there might be a connection there. We also saw that low pHs caused a lot of false positives. which really just goes to show that there are a lot of things to account for if you want something that's very robust and works across many different sample types. So we moved on to round 2. We essentially started over from scratch, this time keeping in mind that we wanted to build an assay that should work with many different types of patient samples. And I'm just going to show you an example of that here. So what you have on this side is, again, the signal intensity in across 20 replicates of samples containing no venom, and then these different sample types. So what you can see here is that we more or less succeeded, like at least across these 20 replicates with no venom in them. We don't get any positive samples for all these different types of sample types. Conversely, when we do add venom, we do get positive samples, like we should. But something that's very interesting is for each of these samples you see displayed here, we actually added the exact same venom concentration. But for instance, we just get a much stronger signal when testing that particular venom concentration in heparin than when testing it in a specific type of serum. All of this just goes to show, again, like there is a lot of things, a lot of like technical challenges you have to deal with in these development processes, which is equally fun and frustrating, I would say. Okay, but of course, technical challenges aren't the only thing in the way of developing diagnostics. There are also implementational challenges. So one thing I'm thinking of here is that, at least in my opinion, we probably will reap the highest benefits from diagnostics when they can be paired with effective and affordable treatment. But in some parts of the world, treatment just isn't readily available, and that might diminish the effect of diagnostics. So that's an implementational challenges. There are also other types like cultural challenges or technological adoption by relevant stakeholders. So there are a lot of things you could talk about with this. And finally, and this kind of goes back to what Juliette was just discussing a little bit, there are funding challenges. So Polysecure Research, where Juliette is from, actually published these very interesting numbers, which shows that the vast majority of funding for snakebite envenoming go to different kinds of therapeutics and treatments, and only 5 to 9% go to diagnostics. And personally, I think that's a little bit of a shame, because diagnostics typically have a shorter time to market than treatments, so they could potentially benefit patients sooner than new treatments. And I think that's really relevant, especially given that the World Health Organization has published this strategy to reduce the morbidity and mortality from snakebite by 50% by 2030. And I really think that's something diagnostics could potentially help with. But yeah, anyway, that was all I had to say, and I'd be happy to take questions now.